All right, hello, welcome to chapter 7, which is going to be all about protein function. So in this chapter, we're going to go over a number of different examples of protein function. In this first lecture, we're going to talk mostly about oxygen storage and transport. And in the other lectures, we'll talk more about muscle contraction, as well as the immune response. And I just want to highlight over here some of the principles that we're going to be seeing over and over here. So we are really going to be focusing on the diversity of structures that are available within proteins and among proteins, and how that diversity in structure really promotes this diversity of different functions that we're going to talk about, and how a lot of times structures are directly related to functions, and as well as the interactions among different proteins. And so all of these things are tied together: function, structure, and interaction of proteins and other groups. So let's get talking more about oxygen storage and transport. So first we'll talk about myoglobin. Myoglobin is a protein that binds to oxygen in the muscle tissues. And myoglobin releases oxygen under low oxygen conditions, for example, when you're holding your breath or during intense anaerobic exercise, when you're unable to deliver enough new oxygen to the tissue. And so myoglobin's job is really to facilitate the diffusion of oxygen in muscles. So myoglobin binds oxygen and it releases it in order for it to pass through muscle tissue. And this is able to increase the effective oxygen concentration deep within the muscle, where otherwise it might have more difficulty to penetrate. So let's talk about the structure of myoglobin and how that relates to its function for binding and releasing oxygen when necessary. So myoglobin was the first protein structure that was determined, and as you can see here, it's primarily made of alpha helices that are connected together by these sort of disordered regions. And you can see here shown in yellow is a heme group, which is a prosthetic group that is the actual carrier for the oxygen. So let's talk a little bit more about that heme group. So at the center of the heme group, we have an iron 2 ion, and that itself is what holds onto the oxygen molecule, which is shown here above the heme group. The large hydrocarbon ring structure surrounding the iron is called a porphyrin ring, and it itself is stabilized within the myoglobin protein by a couple of different amino acid residues that surround this whole prosthetic heme group, which we can see more easily on the right side of this slide. So underneath the heme group, we have one histidine, and you can see here on the left how the imidazole group that's part of that histidine side chain directly interacts with that iron ion. And that one is called the proximal histidine. There's also a distal histidine that's above that porphyrin ring that interacts more with the oxygen molecule that's bound to the heme. And then on the sides, we have two hydrophobic residues. We have a valine and a phenylalanine. So all those things together help to stabilize the heme group as well as the oxygen that it's carrying in myoglobin. So let's talk more about the thermodynamics of how oxygen binds to myoglobin. So myoglobin essentially has two different states, right? You can have myoglobin on its own without oxygen. When you add oxygen to it, you have this oxygen-carrying myoglobin. And you can also have the reverse, where you have myoglobin with oxygen bound that then dissociates into an unbound myoglobin with a separated out oxygen that is released. So to talk more about this process, we often refer to what we call the dissociation constant, or the KD. And so the KD is essentially our separated components, our myoglobin with our free oxygen in the numerator of this constant, and in the denominator we have our myoglobin with bound oxygen, and that's shown here in this formula. So our dissociation constant here has our sort of separated components or dissociated components in the numerator and our bound components in the denominator. And another way that's often helpful to think about myoglobin is in terms of its fractional saturation, or its YO2. And this refers to how much of the total myoglobin is bound up with oxygen. And so the way we can express that mathematically is also with a fraction. In the numerator, we have our bound myoglobin. And in the denominator, we have our total of bound plus unbound, so our total myoglobin. And we can actually combine these two constants here in another way to come up with a new way to express our fractional saturation, which is over here. And in our numerator, we have our pressure, our oxygen partial pressure, and in the denominator we have our dissociation constant, again with the partial pressure of oxygen. And so this helps us to more easily think about the relationship between the dissociation constant, the fractional saturation, and the partial pressure of oxygen that's available in the tissue. So let's think about what that means graphically in terms of when myoglobin wants to bind oxygen and when it might want to release oxygen. So here on the left, we have a picture of our fractional saturation expression or equation, and we can draw that as a curve. And this curve, as we can see in this graph, is a hyperbolic curve. And an interesting point we'd like to see that's pointed out on this curve is at our halfway point of our saturation, which is 0.5 on the y-axis out of 1. So that's 50% saturation. And if we drop down a vertical line there to the partial pressure of oxygen, we are somewhere around 2.8. And that is actually equivalent to the KD. So it's helpful to think about here when we have what it really means functionally for our dissociation constant. So it's helpful to remember if you have a high dissociation constant, that means that you will have weaker binding, because if you recall from our definition of the dissociation constant, the numerator of it contains the dissociated components. So the higher the KD, the more weakly it likes to bind. So let's go on and think about what this means now for another similar but different protein called hemoglobin. So hemoglobin is similar to myoglobin in that it binds and releases oxygen, but hemoglobin is a little different. So myoglobin that we were just talking about binds and releases oxygen in the muscle and the tissue, whereas hemoglobin's job is to bind oxygen in the lungs and then carry it through our blood to the tissues so that it can then be used during our metabolism, during our cellular respiration. So if we follow this little chart around, if we start, we can inhale oxygen from the air. That oxygen then gets bound to hemoglobin, which we see over here, our bound hemoglobin. Our bound hemoglobin then passes through the blood, where it then releases the oxygen. The oxygen then gets passed into the muscle onto myoglobin. The hemoglobin itself, once it releases its oxygen, can then travel again through the blood back to the lungs to pick up some more oxygen. But let's follow our oxygen. So our oxygen bound to the myoglobin. Now we have our bound myoglobin. We go through cellular respiration. We use that oxygen to then create carbon dioxide and water within our muscles. Now that carbon dioxide then travels back through the blood, and we are over all the way back to our lungs, where we are then able to exhale the carbon dioxide and water. And meanwhile, our unbound hemoglobin, like we mentioned earlier, traveled also back through the blood so that it's all ready to pick up some new oxygen when we inhale it again. And this cycle sort of continues over and over. And we'll revisit this idea later as we go through the slides. So let's move on from here. So let's talk more about the structure of hemoglobin. So hemoglobin, as we've mentioned before, is made of 4 different subunits, and each one has a prosthetic group, that heme group. You can see those in red here on the left. And it's fairly easy to see from this diagram that there are 4 different subunits, and it's maybe less obvious that there are 2 types of subunits and you have 2 of each. And so in the case of hemoglobin, we have 2 alpha subunits and 2 beta subunits. And just in case anyone is confused, the alpha and beta here do not refer to the secondary structure within those subunits. It's just a shortcut for one type and another type, an alpha and a beta. As we mentioned before, for myoglobin, the actual secondary structure present in any of these subunits is actually mostly alpha helices, because the structure of the subunits of hemoglobin are very similar to the structure of myoglobin, which contain only 1 subunit, while hemoglobin has 4 of these myoglobin-like subunits. And so, so we have 2 alpha subunits, 2 beta subunits. So we have a heterotetramer. And you can see here on the right, we can see more of these, the more detailed structure looking at these alpha helices. And you can also see more clearly where the heme groups are. So we have in our alpha subunits, they're over here. In our beta subunits, they're over here. And in the, so that's all I want to show here for now for the structure of hemoglobin. But let's talk a little bit more about the conformations. So we've discussed a little bit in class how a lot of proteins have some amount of variability, a little bit of variability within their tertiary structure when they're in solution, depending on their environment or other factors, and hemoglobin is no exception. So there are 2 conformations that hemoglobin can have, the T state, which is shown, on the left, and the R state, which is shown in red. And so the differences are subtle, but if you look at this closely, you can see that there are some shifts in some of these alpha helices that change how they interact with one another, and it moves around some of these alpha helices very slightly in shape. One of the more obvious consequences of these shifts between the T state and the R state is actually in the center, and where you can see in the T state there's sort of a larger space, versus in the R state there is a smaller space for oxygen to bind. And so this can make a difference whether your hemoglobin wants to hold on to oxygen or whether it wants to release oxygen, which state it's in, if it's in the T state or if it's in the R state. So let's talk a little bit more about that, how oxygen, just to go back, there are a lot of different factors that can affect whether hemoglobin is currently in the T state or the R state. And one major factor is actually whether or not oxygen is bound to one of the subunits. So oxygen binding can actually change that structure. So we discussed before how the difference between the T and the R state can be some of these relatively subtle shifts in the helices. And so we can see in this picture, when we have, on the bottom we have our heme group, and when oxygen is bound to our iron, there's a really small shift in the angle and the location of the porphyrin ring, which moves around where this histidine is in space in the middle here, this distal histidine, maybe it's the proximal histidine, and so that shift in this histidine, which is part of an alpha helix, when you move that histidine, it moves the entire alpha helix into a slightly different location. And when you change that location, it shifts how that alpha helix interacts with other alpha helix, both within that same subunit and in neighboring subunits. And so that can this shows us more locally what oxygen is doing to the alpha helix that's sort of touching where the oxygen is. But we can also see these differences in structures that are not local to one another. So this is showing alpha helices on two different subunits, right? We have the alpha 1 and the beta 2 subunits. And so you can see this alpha helix on this alpha subunit has a proline, a threonine, and two threonines. And our beta subunit, we have a histidine and an aspartate. And you can see on the left, the histidine is sort of right next to the proline, and the aspartate is sort of in between the two threonines. On the right, we have this shift that's occurred from the T state to the R state, right? And so now our histidine is actually in between the two threonines, and our aspartate has shifted all the way above there. So these two alpha helices that are actually on entirely different subunits have now shifted where they are in relation to one another, which goes on to overall affect the shape of the hemoglobin, which goes on to affect the hemoglobin's function, its ability to bind or release oxygen. So let's talk a little bit more about that oxygen binding. So this graph should look a little bit familiar. So here's our curve from myoglobin with that hyperbolic shape. Where you have this asymptote just below 100% saturation. And for now, when we're talking about hemoglobin, when we want to talk about its relative saturation, we have a similar equation here, except now we have the shape of our graph has changed because now we've added these exponents for n here, and so when we add on subunits, we make this process of binding and releasing oxygen much more complicated. Myoglobin just has one subunit, one oxygen, as a fairly straightforward curve. Hemoglobin has 4 subunits. We can bind up to 4 oxygens, and so this gets a little messier. And so you can see here in the other solid line on our graph is our actual curve for what hemoglobin looks like. And so the shape of the curve for hemoglobin actually has a little bit more of an S shape, and so we call it a sigmoidal curve. And so if we wanted to, again, calculate our halfway bound state for hemoglobin, we would be just about here at the 0.5 mark, and if we drop down this vertical line, we are close to about 30 torr, which is very different from myoglobin, which, as you recall, was closer to about 2.8. And so the dissociation constant for hemoglobin is higher, means it's easier, it has weaker binding, right? Which makes sense, right? You have hemoglobin carrying oxygen through your blood to your tissues, where you then want to be able to release that oxygen in the tissue so that so myoglobin can take over for you, right? So myoglobin naturally is going to bind more strongly to oxygen in general than hemoglobin does. And of course, this depends on the pressure, right? So our halfway point here is right around what we have for our pressure in our veins. That's our hemoglobin that's returning to the heart that has dropped off its oxygen, essentially, versus when we want to look at our arterial pressure, that's a little higher. That's about 100 torr, as we see on this graph. And so we want to have a fairly large, it's helpful for hemoglobin to have a fairly large difference in the amount of saturation between the veins and the arteries, right? Because you want to pick up oxygen in the parts of your blood that are more oxygenated, and you want to drop off oxygen where it's not, right? And so the shape of this curve allows hemoglobin to have about half bound at venous pressure, and if we follow this chart up at the arterial pressure, we go up to about 90% here at arterial pressure. So we have a pretty big difference of about 40% difference. And if it's helpful to imagine, we can compare it to this dotted line curve. So this dotted line is sort of an imagining of what a normal hyperbolic curve would look like if hemoglobin were to have this hyperbolic curve rather than the sigmoidal curve that it does truly have. And so you can see here that the difference between where the asymptote is located for this theoretical hyperbolic curve is relatively low. It's below 80. It's maybe 70, 80%. And so the difference there between the 50 and the 80% is a lot lower in this theoretical curve than it is in this real-life sigmoidal curve. And so why is that? It's because we see some cooperativity in oxygen binding between the subunits of the hemoglobin protein. So like we mentioned, that 4 different subunits of hemoglobin are each able to bind an oxygen. And it's helpful for if one subunit, or the way that it works is once one subunit binds to an oxygen, it becomes much easier for the other subunits to also bind to oxygen. On the other side, if once you start releasing oxygen, it's also easier for the other ones to start releasing oxygen. And part of that has to do with the differences in pressure that we just talked about, but also has to do with the T and the R state of hemoglobin. So when we think about the T state is more likely to release oxygen, and the R state wants to bind more tightly to oxygen. So when one subunit binds to oxygen, we have that shift to the T state. Sorry, we have that shift to the R state from the T state, and then once that one subunit changes, the other subunits, because as we talked about, we have this relationship when one subunit changes its conformation, we see those shifts of those alpha helices in relation to one another. And so if one subunit goes from the T to the R state, the other ones will also follow. So it makes it easier for all 4 subunits in hemoglobin to sort of affect one another. So what other things can affect the state of hemoglobin, whether it's in the T state or the R state, besides the actual binding to oxygen? One of these regulators is called bisphosphoglycerate, or BPG. So BPG is an allosteric regulator of hemoglobin. So first of all, what is an allosteric regulator? So allosteric means that this component is able to bind to a protein or an enzyme somewhere away from its active site. And even though it's not binding on the active site, it's still able to change that enzyme's activity in some way. And so BPG is one of these things. So it binds to hemoglobin somewhere that's not in the active site. And you can see a depiction of that here. It sort of binds in the middle of all those subunits in that central cavity. And so when it does that, it stabilizes the T state of hemoglobin. And by stabilizing the T state, this overall reduces oxygen binding, because like we mentioned, when hemoglobin is in the R state, then that's when it wants to bind oxygen more tightly. And so BPG, by reducing oxygen binding, allows for that cooperativity that we just discussed, where if one subunit wants to release oxygen, then they will all do it together. And one reason that BPG is sort of useful to have this allosteric function is because BPG is one intermediate in the process of metabolism, of taking in nutrients and using them for cellular respiration to create energy. And so oxygen is a requirement for this creation of energy. And so when we have the production of BPG, we want to then, it makes sense for hemoglobin to want to go into the T state and release oxygen, because the cells are going to want to take up that oxygen that hemoglobin is releasing and use it to make more energy. So let's talk about another effect on oxygen binding in hemoglobin, and that is the Bohr effect. So the Bohr effect overall essentially is defined as how the pH can affect oxygen binding to hemoglobin. And so it does that by this idea where you have these salt bridges. So we have, in a lower pH, we're going to, on these basic amino acids will have these charged states. And so, and then we'll form these salt bridges that we see on the lower part of this figure. And overall, those salt bridges will help to stabilize, again, the T state of the hemoglobin, which will overall decrease oxygen binding and, again, allow for cooperativity. And so, again, this is mediated by these ionic interactions that we see drawn here below. So what does that mean graphically? So that actually means that our curve for oxygen binding for hemoglobin is actually going to shift depending on the local pH. And so at the lungs, you might have one pH that's maybe a little bit higher, versus in more oxygen-starved tissues, you might actually have a higher pH. And so this will affect exactly where that 50% bound part of the curve lays, and so it will affect whether or not hemoglobin wants to hold on to oxygen or release oxygen. And so overall, when you have somewhere where you have a lower pH, we're going to raise our KD, right, by shifting to the right. And so that's going to increase our KD by this curve shifting to the right. And so we're going to have a more dissociated state. We're going to release oxygen. And that's often, like I mentioned, in more like oxygen-starved tissues. On the other hand, if we have a higher pH, our curve is shifted to the left, which will actually decrease our KD. This will allow for tighter binding. So let's think about what this means. Let's break it down a little bit. So we're talking here and now about our Bohr effect, which is, again, pH affecting the state of hemoglobin and how that works with carbon dioxide. So when we're in a low oxygen environment, such as capillaries or any kind of oxygen-starved tissue or area in your body where your hemoglobin is going, we have this low oxygen level. And so when we have low oxygen, that means we have higher carbon dioxide, because usually when you're using up your oxygen, you're making carbon dioxide as a byproduct along with water, like we showed before. And when we have high carbon dioxide, once that dissolves into an aqueous solution, that will give us high carbonic acid. And so high carbonic acid, then a proton can dissociate, right, which lowers the pH. This is an acidic sort of environment. And so overall, this is going to decrease oxygen binding and allow us to release oxygen, because again, we talked about with the Bohr effect that low pH shifts the hemoglobin more into that T state, which is going to help it release oxygen. This is in contrast, we see the opposite of this now in the lungs or any other high oxygen type of environment. So here we have high oxygen. Now we have this more binding of oxygen on the hemoglobin. And so we'll have the hemoglobin can have this release of protons, and overall that will lower the pH. Lowering that pH will then convert bicarb into carbonic acid and allows us to release dissolved carbon dioxide from the blood, which we ultimately then breathe out. So let's talk a little bit more about how carbon dioxide itself can stabilize the T state. So not only does carbon dioxide have this effect on the pH that we just discussed, it also can directly bind to a part of the hemoglobin protein and directly stabilize the T state. So let's discuss that. So we have this little diagram here where we show the N-terminus of one of the subunits of hemoglobin. When carbon dioxide is around, it can actually bind to the N-terminus and form this carbamate. And so when this reaction is more likely to happen in the T state. And when it does happen, it again stabilizes hemoglobin in the T state. And once again, the T state is going to allow hemoglobin to then release more of its oxygen. So aside from decreasing the pH, which stabilizes the T state, which we just talked about, we also now are seeing how carbon dioxide can form these carbamates with the N-terminus and also further stabilize the T state. So let's look at all of those things together. So we talked about how Oxygen binding can affect the state of hemoglobin. We talked about how pH can affect it. We talked about how carbon dioxide can directly affect it. So let's bring all of those things together. So this diagram looks a lot like the diagram that we saw several slides ago, but now we have some more details filled in. So we start like we started before with we breathe in, right? We breathe in oxygen, and now we have hemoglobin that has traveled to the lungs through the blood, and it's going to take up some of that oxygen. So we have this bound hemoglobin bound with oxygen. The hemoglobin is going to carry that oxygen through the blood and bring it over to the muscle or any tissue that needs oxygen. And so once hemoglobin arrives at the tissue, it's going to release its oxygen, partially because of that change in partial pressure. So in this high oxygen environment, hemoglobin wants to bind the oxygen. In this low oxygen environment, hemoglobin wants to release the oxygen. Once it gets released in the muscle, that oxygen is going to bind to myoglobin. And then eventually that myoglobin can be used by the tissue for cellular respiration to create energy where it's needed, to keep your muscles going, right? And so, and one of the byproducts of respiration is carbon dioxide and water. And so now that carbon dioxide is going to re-enter the bloodstream, and it can, by itself, it can convert to carbonic acid, but that's a very slow reaction naturally. So we conveniently have this enzyme called carbonic anhydrase, which with the help of water will convert carbon dioxide into carbonic acid, which, like we mentioned before, will a proton will dissociate and we'll get bicarbonate as well as a proton. And so that proton can then act to protonate the hemoglobin, and that protonated hemoglobin is able to then travel through the blood. Once you get back to the lungs, now we have this different environment, right? And so the hemoglobin is going to then release the proton. That proton can then combine back with the bicarbonate that's been traveling through the blood. Again, with the help of carbonic anhydrase, we can reform that carbon dioxide, which comes out of solution in the lungs and gets exhaled along with carbon dioxide and water. So that essentially sort of summarizes all of the different, or a bunch of the different concepts that we've talked about here in terms of hemoglobin, when it wants to bind oxygen, when it wants to release oxygen. And so now just to close things off, let's talk a little bit more about a clinical consequence of what happens when something gets changed in hemoglobin, and that is a disease called sickle cell anemia. And sickle cell anemia is actually caused by a single point mutation that we see in hemoglobin. And so when we see this point mutation in hemoglobin, we call it a hemoglobin S. And so what happens in hemoglobin S is that we have this amino acid change that we just mentioned, right? So in the beta chain of this beta subunit of this hemoglobin has a mutation from glutamic acid to valine. And so hopefully you know by now this is a very non-conservative substitution, right? Because glutamate is negatively charged, it's acidic, and valine is nonpolar and neutral. And so, this particular amino acid is located on the surface of this beta subunit. And so this can have a pretty big effect, as you can see down here, where now we have this valine. It interacts with this hydrophobic pocket on another subunit that we see over here with a phenylalanine and a leucine. And these hydrophobic kinds of interactions ultimately make this hemoglobin S polymerize into these fibers that we see on the right. And so it's these hydrophobic interactions between the valine, the phenylalanine, and the leucine among different subunits of hemoglobin that are deoxygenated, and they form these long polymers, these long fibers. And if they're long enough, they can start to both change the shape of the red blood cell by elongating it, essentially, and then potentially eventually lyse or break open the red blood cells, which is a really big problem because now the red blood cells are, one, no longer able to carry oxygen to your tissues because they're broken, and two, you have this maybe buildup of debris or even of whole blood cells. They can build up and cause clots and ultimately cause a huge disruption in your body, including giving you joint pain and other symptoms. It's really, not not a fun disease to have, obviously, of course. And so this is, on this slide, we have just sort of a picture of, these are our normal red blood cells. They're round, they're almost sort of donut shaped. But in sickle cell anemia, we have this elongation of these red blood cells into what looks almost like a sickle shape. That's where the name comes from. And it's because of these long polymers of hemoglobin S. They make these long fibers, and eventually they can actually break open the red blood cells, which you see here, which again causes a lot of varied symptoms for these people who have this disease. One maybe interesting thing to note about sickle cell anemia is it has this connection to malaria. So something that people noticed a while ago is that people who are heterozygotic for this point mutation for sickle cell are actually protected against malaria. And so you can see that geographically in regions of the world where there's a lot of malaria, there's also a selection for the sickle cell gene because, again, if you're heterozygotic, you actually have this protection against malaria, which is another deadly disease. And so the thought behind this is, as you might know, malaria is caused by this parasite that lives part of its life cycle inside of red blood cells. And so there's some thought that there's something about this maybe partial polymerization or this partial sickle cell nature of these red blood cells that somehow prevents the malaria parasite from colonizing inside of the red blood cells instead. So that's definitely some food for thought. And another thing to note here is the treatment for sickle cell anemia, which is hydroxyurea, and the structure of that is shown below. And so the exact mechanism for how that works isn't known, but this helps the red blood cells maybe from clotting. Maybe it helps to somehow straighten out some of those proteins that are clumping up or polymerizing. But overall, these are just sort of some interesting clinical connections to note that are related to hemoglobin and its ability to find and deliver oxygen at the lungs and at the tissue. So now hopefully you are ready to answer these oxygen storage and transport practice questions.